Hello, we would like to present our BBSRC Follow-on Fund project entitled Commercialisation of a Therapeutic Bacterial Vesicle Engineering and Manufacturing Platform. My name is Dr. Richard Kelwick and I'm a Senior Research Associate in Professor Paul Freemont's group.
Hello, I'm Dr. Alexander Webb, a Senior Research Associate in Professor Paul Freemont's lab. Bacterial outer membrane vesicles, or OMVs for short, can be used as vaccines to protect humans and animals against a range of diseases.
Thank you, Alex. That is why we are developing our Omega platform to make it easier to engineer and manufacture OMV-based vaccines, including those to help protect poultry against avian pathogenic Escherichia coli, or APEC for short.
Every year, APEC infections devastate poultry farms, causing stunted bird growth, fewer eggs, and can even wipe out entire flocks. Avian pathogenic E. coli infections in chickens, turkeys and ducks impacts over 30% of US commercial flocks and globally causes hundreds of millions of dollars in industry losses every year. APEC-infected birds suffer from severe diseases that veterinarians call colibacillosis, leading to systemic health issues and mortality rates of up to 60%.
Because of this, that's why we are developing our Omega platform to generate these bacterial outer membrane vesicles. A part of this as well is, despite the huge impacts that APEC has on the poultry industry and human health more widely, the current solutions to protect poultry against APEC are ineffective. For example, antibiotics are decreasing in use due to, of course, antibiotic resistance. There's also a general trend in terms of regulations and other policies minimizing their usage. But also farmers themselves know that poultry treated with antibiotics can have side effects which impacts their health, their meat production and also their egg production. Unfortunately, the currently widely used live attenuated vaccines, such as those developed by Zoetis and Chemin, whilst fairly effective, are not always fully effective, especially against changing strains and for cross-strain protection. Additionally, farmers also want rotation products. This is because farmers are rightly concerned about resistance emerging against either vaccines or antibiotics, and so they want different solutions to protect their flocks moving forwards. Therefore, our proposed approach is to make it easier to develop OMV-based vaccines, and so we have developed our outer membrane vesicle engineering and manufacturing, or Omega platform, to make it easier to create better and more scalable methods for manufacturing new kinds of therapeutic or vaccine OMVs. In our project, we are using Omega to design, engineer and manufacture across different scales a panel of candidate OMV-based APEC vaccines. We will also characterize our vaccine candidates and use in vitro screening to determine their effectiveness. And of course, we want to expedite a responsible translational pathway for our Omega platform. And we are pleased that we have collaborators and supporters in our project, including but not limited to Symbicity, the London Biofoundry in Imperial College, and the Future Biomanufacturing Research Hub at the University of Manchester. So, our Omega platform. Our Omega platform allows us to create bacterial outer membrane vesicles that are engineered from standardized bacterial strains. They allow us to display multiple vaccine antigens, and we can also have designer protein cargoes. Our platform is modular and standardized, meaning we can create many vaccines from the same platform and also many kinds of therapeutics. We believe our approach is novel in that we can enrich for our engineered OMVs, which distinguishes from other technologies which often isolate a mixture of OMVs or EVs produced by different cell types. And our approach is also scalable. And as part of our project, we want to go from our current small scale in the lab to a higher scale using a bioreactor system. And in discussion with Imperial Enterprise, we believe our approach is patentable. There are core innovations as part of our platform. In terms of our future vision and expected outcomes, As part of our project, of course, we want to demonstrate our Omega platform and its ability to create many different therapeutics and vaccines. Of course, our focus is on the development of poultry vaccines, and so we want to demonstrate their engineering and manufacturing at different manufacturing scales, and to test our candidate poultry API vaccines for their efficacy in vitro. In terms of long-term impact, we want to continue our engagement with industry, and this builds upon our participation in the BBSRC iCure program. And we believe that we can file a patent for our Omega technology, and we have support from Imperial Enterprise to do this. And in the long term, we believe there's scope for licensing of our technology and platform and/or potential spin-out.